The topic for the present video is process related impurities in the pharmaceuticals. These impurities are not the degradant impurities. That means generally these impurities are not those impurities which get increased during storage and shelf life. These impurities come from the manufacturing process or synthesis process of the API, and these are controlled at the initial stage by the API vendor. And generally these process related impurities are not studied by the drug product manufacturer. So I welcome all of you to the present video on process related impurities, and in this video I have tried to cover most of the information related to these impurities. See, as we know, the impurities are of different types like process related impurities, degradation impurities or degradants, and other impurities like residual solvents, elemental impurities or heavy metal impurities, nitrosamines, genotoxic impurities are there. Out of those, we will see the process related impurities. These impurities generally origin from the manufacturing process or synthesis or ROS, that is route of synthesis of the API, and these come from the starting materials like raw materials, intermediates, reagents, solvents, catalyst used for the manufacturing of the API or drug substance. And generally these process impurities are controlled by the API supplier or API vendor. These are identified and controlled at the development stage only. These are studied at the synthesis stage of the API during research and development, scale up, and the exhibit batches of the API. These are controlled by the drug substance manufacturer through the validation batches, through quality control and purification of the API. These process impurities are required to be controlled at the validation stage, and the control strategy is required to be made to control the process impurities through the manufacturing process optimization, controlled parameters, and validation of the critical stages. Then the process impurities are controlled and specified in the DMF. Detailed investigation of the all impurities is must, and it is required to be provided in the DMF file. These process related impurities are also captured in the API COM. These impurities are generally not included or monitored in the drug product. For example, if you have taken a drug substance, if it contains 5-6 impurities, then the drug product manufacturer is not required to study all those impurities in the stability. Only the degradant impurities are required to be studied. And the basic difference between the process impurities and degradant impurities is that process impurities are not the impurities which get increased on storage or during the stability. These are not the degradants. While the degradants are those impurities which get increased on stability study, during storage, during manufacturing, or these are formed because of the API degradation. Process impurities are not the part of degradant impurities. And if the process impurities are getting increased, for example, suppose you have API, in that impurity A, for example, let's say it is a process impurity and it is getting increased on stability. So it is process impurity, also it is a degradant impurity. And if the impurity is process related, and if it is also getting increased because of degradation, then it is very important to control that impurity and include that impurity in the finished product specification. All the degradant impurities are required to be included in the finished product specification. The references for process impurity specifications are ICH guidelines, like ICH Q3A is there for drug substance, and chapter in the USP is there 1086 for impurities in the drug substance and drug products. So from these literature you can get the exact idea about the impurity specification. Process impurities are required to be investigated, identified and controlled in the drug substance. And these impurity list, along with their nomenclature, structure, and the root or origin of that process impurity is required to be given in the DMF. If it is a process plus degradant, then that is required to be included. These are required to be studied in the stability study during research phase of the API or drug substance, and if not found to be degradant, then can be considered as control at the API stage, and then these are not studied further. These can be predicted based on the route of synthesis and the raw materials used and the intermediates formed in the route of synthesis of the API. The process impurities are to be investigated in the research and development phase and are less focused in the long-term stability studies of the drug substance and also the drug product. Process impurities can be controlled through the process studies and optimization, and these are the part of the impurity profile. You cannot say that it is a process impurity, so we will not include in the impurity profiling. See, impurity profiling should be there for all the types of impurities, whether these are degradant or whether these are process related. So specification as per ICH Q3A, impurities in the new drug substances, I have included here, and these are given based on to the maximum daily dose. If the maximum daily dose is 2 grams or less than that, then a reporting threshold will be there as 0.05%, identification threshold will be 0.1% or 1 mg per day, whichever is lower. Qualification threshold will be 0.15% or 1 mg per day as per the maximum daily dose, whichever is lower. If the maximum daily dose of the API is above 2 gram, then reporting threshold will be 0.03%, identification threshold will be 0.05%, and qualification threshold will be 0.05%. So these are the specification requirements or specification criteria for process related impurities. Generally, the impurity related questions are asked in the interviews, and being a pharma professional, we should know about these process related impurities. Also, always remember that if a process impurity is also a degradant impurity, then that impurity is required to be included in the finished product specification, and it is required to be monitored throughout the stability studies and shelf life. You cannot omit that impurity, as it is a process related impurities come degradant impurity. That means that impurity is both. It is also coming from process, and also it is getting formed due to degradation of the API. If the impurity is purely process related impurity, then it is considered that it is controlled at the API stage, and it is not required to be studied further in the drug product manufacturing and stability studies. Be clear on these terminologies and always remember these important points. Thank you for watching the video, and please do like, share and subscribe to this channel. Share these videos to your friends and colleagues. Thank you.